afternoon, everyone. But before that, I would like to congratulate to the organizing committee for having this, the first BioNano seminar there. Dr. Nick. Okay, this is such a very big platform for us to meet up, catch up, okay, see each other, hear about each other's work, and then perhaps we could share whatever that we have along the journey during your time here. Right. So I'm not going to introduce you what is wound healing, what is the definition of that, because everybody knows that. I think at least some of you. Farida would know. Yongchi, Pedro, and Haris. These guys are in this area as well. Okay. So what I'd like to highlight here is on the development of biomaterials for wound healing agents. So biomaterials have been reported to have lots of bioactivity, and one of them is the wound healing activity. And it is not surprised that some of the biomaterials activity have been available in the market, which means that they have been through a lot of work. From the lab experiment to the clinical trial. Some of them may escape clinical trial and then eventually be available in the market. So from the bench to bench side, if I would ask you, what happened in between? Yeah, so mainly experimentation, which involves a laborious work, time, not only that, but also uses a lot of money. So we, as a researcher, we have to acquire for funding to have this kind of research to be done. And on top of that, this is also accompanied with some sort of emotional feelings such as frustration, happiness. Bila you dapat results, you happy, and then bila tak dapat, you sedih, And then you have to be resilient, and you have to be persistent in your research. So this is all what happened in between lab experiment and for the product to be available in the market. Okay, so I'm not going to translate this into. So I'm not going to leave you with only this cartoon. So today we're going to look at what actually happening in between these cartoons. So the first experiment, the first questions that normally we have to ask ourselves before we embark into any experiment associated, related to wound healing is that, what is the appropriate dose? Okay, so before you do any wound healing assessment, so these questions need to be addressed. So the first experimental method should include determining the safe dose of your materials. So these can be done by the in vitro studies, i.e. cell culture, which normally people use cells such as fibroblasts, keratinocytes, and other types of cells. So fibroblasts and keratinocytes is the main cells available in the skin microenvironment. Keratinocytes is the most abundant types of cells, and that's why normally we tested on keratinocytes. So the experimental involves cell viability assays or cell proliferation assay, where we normally look at the metabolic activity. A lot of people using the most classic methods involving the use of MTT reagent. Other than that, we also have the MPS or XTT. This is mainly looking at the metabolic activity, where we want to measure the presence of viable cells by calorimetry. And moving on to a bit more sophisticated methods, not sophisticated, but because it produces pretty images. Okay, so we can determine cell viability by using the live dead staining. So this is where we introduce dyes, specific dye that can bind to dead cells and live cells. For example, in this image here, the live cells can be visualized in green color, while the dead cells can be visualized in red color. So we can count how many live cells, how many dead cells, and we can kind of like have the exact ratio of the live dead cells in following one experiment. And we have also flow cytometric analysis where we could stain our cells with PI. PI is a dye that can differentiate between the dead cells and the live cells, where the live cells will be stained for negative PI. Okay, so from this we can actually determine the percentage of cell viability AC. People also looking at the rate of proliferation, okay, by doing some DNA measurement. This is usually can be done by various types of kits, which what we have in our lab is, for example, the cytochrome dye, which can directly bind to the DNA, as well as the BRBO uptake assay. Okay, so the answer from this experiment will lead to the whether or not the materials that we are going to use in our experiment, whether it is safe or harmful to the cells or unsafe. Okay, so the answer would be yes or no. So if it's safe, then does it stimulate wound healing? Or if it's unsafe, then we have to retest the dose, which means that we need to reduce the concentration of materials used in the experiment. Alright, so once we've got the answer for what is the safe dose for our materials, then we normally move on to the second experiment, which involves the in vitro study. Now the questions remain: does it stimulate wound healing? Okay, so in order to answer these questions, we firstly perform the in vitro models, which is based on the scratch moduli format. This is a 2D format where we will culture cells such as the granulocytes and fibroblasts again using the condition that we established in the first stage, and then seed the cells, and then in a monolayer, and then we are going to introduce scratch deliberately, okay, by mechanical wounding. So there are various types of mechanical wounding that can be done, either by as simple as pipetting, or you could also use stamping using a metal here, for example, where we can create a round shape of wound in the center of the well. We could also use electric stamping, ini adalah hot stamping, which this stamp is actually very hot, causes the cells to die, and thus creating a wound. And this one here is electric mechanical wounding, in which underneath the plate has some sort of like metal connected to the electric, where we will introduce some sort of like vibration that eventually causes cells to detach, and hence creating wounds. And finally kita boleh buat wound melalui laser optic. This variation in mechanical wounding, okay, however, may cause some inconsistency in getting a reproducible results. Sebab, for example, scratching using pipette tape normally will give you a wound that has not similar in size. But of course, by practice, by experience, we may increase that skill. Okay, and other than that, we could also use the commercial inserts, which we could put silicone in the well, and thus creating a wound by these silicone, by these commercial inserts. But the drawback is that it is quite expensive. And then we're going to do some treatment using the dose that has been determined in the previous experiment. And then we are going to monitor the wound closure by either taking an image at different time interval, which normally we done in our lab, or we could use the time-lapse camera. Unfortunately, we don't have this kind of camera setup in this faculty. Whatever the approach used, we will obtain serial images at different time points, and we're going to analyze it by using various software for image processing, One of the famous one is the image. Okay, we could also use the image processing toolbox by the MATLAB. I think UTM has subscribed MATLAB software, so I'm not sure whether this plugin is available, but we could explore later. And then some of the other freely available software is called the TS Scratch, the T Scratch, and multi-cell experimentation, multi-cell seq. So this image will be processed, okay, and be analyzed for its wounding healing capacity. So this work here is being done by our friend here, okay, and this has been published back in 2019, where Harris has been doing some in vitro wound healing experiment in which he has acquiring the image at every 4 hours interval, and then he's process the image and measuring the wound gap following any treatment that he used in his experiment. The numbers that is obtained from this analysis can be also translated into a graph, okay, so that the reader can follow what actually happening, which allows you to do some stat analysis. Right, so other than in vitro scratch assay, there are a few other experiments that can be done for in vitro migration assays. One of the most commonly used is the Boyden chamber assay. This is like the trans-well migration assay, which you have like a set of well with some insert, okay, and you're going to seed the cells inside the insert and then immerse in the media containing your test material. From there, any cells that are able to migrate will cross through the membrane, okay, while those that cannot migrate will live in the well, in the insert. And then we're going to remove the membrane and stain for using like a certain dye and count for the cells that are able to migrate, i.e. trapped in the membrane. Right. So that is another way of like determining the migration activity following any treatment of a test material. But what makes in vitro scratch assay is more convenient is that it is very cheap with no extra cost than tissue culture, while the Boyden chamber requires some purchase for the membrane. However, it is still considered as not expensive as the other two methods here. Moving on. Now we've done the 2D format. Again, we would like to ask whether does it stimulate wound healing. If it's yes, then we can test it to a more elaborate in vitro model using the human organotypic skin explant culture. But if it's no, then does it impair healing? And normally you have to review back the study aims. Perhaps the test material that you're using is not possessing any wound healing capacity. So there is no point of you testing forward. This human organotypic skin explant culture is actually a very good model in studying wound healing, as it provides more than 3D format of cells, in which this is actually a human skin that is being collected from surgery, and then of course this was taken by like a consent from the patients. Okay, so and that's why this is like our human thick skin. They cut into pieces and then put it in the well. We're going to culture it. So it consists of all cells that is available in the skin, includes melanocytes, ganglion cells, keratinocytes, fibroblasts, as well as proteins that is required to maintain the structure of a skin. For example, the glycosamin glycans and collagens that give the skin structure. So you could test your test material by culturing these organotypic skin explants in this well, and we can monitor the tissue viability by staining using a certain dye and looking at whether or not the tissue is still viable. We can see here that the tissue is still viable, where this tissue is being stained with the Ki67, which is a specific marker for cell proliferation. So the brownish color there represent a specific region that is positive for Ki67, which means that it is proliferating and it is viable. Okay, right. So this is like another advanced in vitro model that you could use and could be really mimic to the in vivo situation. Now, from this experiment, again, you would ask, does it stimulate wound healing? If it's no, then you have to review back your aims. Okay, perhaps you say that, okay, sampai sini sajalah kita punya activity. Ataupun, if it's yes, then you could embark on another chapter, which is the preclinical studies which involve animal models. Right, so in preclinical studies which involve animal models, we have various types of models that can be used. We could start with as easy as closed wound, kita gunakan implants, ataupun open wound, sama ada kita gunakan incisional or excisional model. So this is the excisional model where we could kita boleh buat wounding using a biopsy punch. Take up the skin, the whole skin thickness out of the animal, and then kita juga boleh buat incisional wound, which is ambil benda tajam and crack secara vertical, ataupun kita boleh create some excisional with splintered wounds, which is gunakan biopsy punch, and kat luar tu kita letakkan silicone splint supaya the wound tu tak bergerak-gerak. So kita buat some surgery. So we did not perform this because it involves surgery and I'm very soft at heart. Of course, in order for you to work with animal model, you have to acquire animal ethics from the animal ethics committee. Then only you would do this kind of experiment. Right, so these are the outcome of your in vivo wound healing activity, where we measure the wound contraction every alternate days, taking a serial photograph, compile it, and we could process it by image processor software, and we could get some measurement out of this. At the end of the day, we could compare in terms of efficacy of treatment in your experimental setup. Other than that, we could also harvest the tissue at a specific time point, and then subjected it for histological analysis, where we could follow the wound contraction. So the arrow here showing the age of the wound, and we could measure from the side to side of the wound area, the age of the wound, and we could translate it to a graph here in length of epithelial gap. So you see that at a later time point of your experiment, the gap becomes smaller, right? Other than that, we could also do some histological analysis as well to look at the collagen deposition. We know that collagen is one of the extracellular matrix provided by the skin to hold the structure of the skin, and therefore collagen is really important and involved in cell migration, in tissue healing. So this is a skin tissue that is being subjected to stain with Van Giesen staining, which is a spatial stain, to visualize collagen, and from here we could use some image processor and translate it into a graph where we could see the production of collagen is increased following specific treatment. We could also do some other in vivo assessments, such as looking at a protein analysis using immunohistochemistry. For example, here we're looking at the proliferation activity in the healing tissue, where we stain our tissue with anti-PCNA, which is a specific marker for cell proliferation, and we count those the green dots here as a positive for PCNA cells, the proliferating cells, and we could translate it into a graphene. Other than that, we could also do some ELISA to look at the expression of a certain cytokines growth factor that involved in the wound healing, or RT-PCR. Right, so from preclinical study, we should be able to answer this question whether or not it is stimulate wound healing. If it's no, then you have to review back your aim, or if it's yes, then you could proceed into using a more complicated model using impaired wound healing models such as diabetic healing, diabetic model, or ischemic model, or obesity, or any wound that is inflicted with infection. Or you could always straight to the entering clinical phase, which has clinical phase 1, 2, 3, and 4, which is a post-clinical studies, which is post-market surveillance studies. Right, so all in all, these are the things that is happening in between, starting from your material that is in the lab, okay, until it is available in the market, right? So this whole process usually takes about 10 to 12 years convention, but then again, with the current technology, then you could cut down time taken for the process, for this process, especially if you could if you could do some in silico analysis, which could reduce the screening process. Obviously, in silico is not perfect. It still requires some backup data from in vitro and in vivo studies. Right, so as a conclusion, in order for you to choose which assessment is suitable for your study, you need to ask that yourself. What experience on wound healing assays is available in your lab? Okay, there's no point of aiming for something that is too high if it's not being established in your lab. Number 2, how many experiments need to be conducted, hence what high throughput is needed. So for example, if your PhD, your project is mainly about synthesizing biomaterials, then you would not need many experiments involving the wound healing experiment. But if your study is mainly on wound healing, then you might need to elaborate more to get more data to support your findings. And then thirdly, which laboratory equipment is available. This is very, very important. Okay, before you start anything, make sure that everything is available in your lab. And then finally, does it currently make sense to invest resources in new equipment or new chemical or new kit to achieve a higher throughput and reproducibility. Alright? So I open to any suggestion from all of you. Thank you very much.